Good afternoon, everyone. You are very welcome to our Meet the Expert Cancer Immunotherapy event here in the Clayton Hotel, Cardiff Lane.
Good afternoon, everyone. My name is Jonathan McCrea. I am thrilled to be here as the MC of this really special event. It's great to see so many of you here as we ask the question, can your immune system treat cancer? Because the answer is a resounding yes, but often it needs significant help. Today, we're fortunate to be in the room with some of the world leaders in cancer immunotherapy, and I'm really excited to hear how they explain the different approaches to how we try and tackle cancer in its various forms. The introduction of immunotherapy sparked immense excitement due to its potential to revolutionize cancer treatment. By leveraging the body's immune system to target cancer, this approach offers a more precise and potentially less harmful alternative to traditional therapies. And one common approach involves enhancing T cells, part of the adaptive immune system, to identify and eliminate cancer cells effectively. And this has led to some really significant outcomes for some patients, but not for all, and that's what we're working towards. This afternoon, we're going to explore the challenges and advancements through 3 panel discussions, and then we'll have a Q&A where you can have your questions to our fantastic panels. Today's event is co-hosted by Breakthrough Cancer Research and the Irish Society for Immunology. We're going to later on hear from Professor Joanne Lysaght on behalf of ISI. But first, please join me in welcoming Orla Dolan. She's CEO of Breakthrough Cancer Research for some opening remarks. Orla.
Good afternoon, everyone. I'm delighted to welcome you here on behalf of Breakthrough Cancer Research, our organisation, and our co-host, the Irish Society of Immunology, led by Professor Joanne Lysaght and Dr. Aideen Ryan, who, as Jonathan said, you're going to hear from a little later. And we're delighted to have you all here with us. For those of you who are not familiar with Breakthrough, we're a medical research charity focused on cancer, and we particularly focus and prioritise poor prognosis and low survival cancers. And we do that with the support of the public and our corporate partners, and we're able to be supporting now about 160 researchers across the island of Ireland and beyond. And hopefully you got a chance to meet some of those researchers during tea and coffee earlier on downstairs, people who are working at the coalface trying to find new ways to treat cancer. Now, I drove up from Cork this morning, and while I was speeding on the motorway, I was thinking a lot about my late father. And because he was the founder of our organisation and he was an esophageal surgeon and a scientist, but he would have absolutely adored being at an event like this, a day where people came together to talk about research, but also how to unlock the immune system to help patients. It would just been in his element having all these people to talk to about it. And he was always passionate about the potential of the immune system and immunotherapy for cancer. And indeed, one of my earliest memories of this is before PowerPoint, and when you had those little slides in the carousel, and he was presenting some data of his own patients. And what he was looking at was, when he took the tumours out of some patients, if you had lots of immune cells infiltrating all of that tumour, those people were alive 10 years later. But when he looked at the tumours of his patients that only had a tiny few cells, or they were only on the periphery, those patients had died from their disease within a year. So even decades ago, he was like, okay, the immune system has a huge role in survival, but how do we get that and how do we unlock it? Fast forward a number of years later, he's involved in a paper called Fast Counterattack, where they identified a target that could be a potential for cancer treatment. So it's why for us, immunotherapy, or the role of the immune system, has been a priority for us for decades. So my own father then lost his life in this young age of 65 to multiple myeloma, and that was over 12 years ago. But today we're going to be hearing about CAR-T, and now they're offering CAR-T to patients with myeloma. So what you see is this huge amount of progress. So the ideas and the potential that he was talking about all those years ago is now a reality. And it's a reality because of many of the experts who will be talking to us today. So that's why we're so excited to be involved in this event. We can't wait to be part of the audience to hear about it. People who are all coming together to say, this is a powerful weapon to go after cancer, but how do we make sure that every patient gets that opportunity? So thank you very much and enjoy the rest of the day.
I'd like to, thanks very much, Orla. I'm really excited about today, and we've been chatting back and forth about the various experts. There's some really, really interesting people with interesting perspectives who are pushing forward this frontier, which is leading to a lot of hope and a lot of excitement. I'm going to welcome up Pat Byrne. Pat travelled here from Moat in County Westmeath, which is a town probably very few of you know, but I actually know it really well because my wife's family picnic happens there every year, and it's about 60 kids running around almost in this field playing football and usually covered in cow dung by the end of the day. So we want to hear from Pat. Pat's going to share his personal experience with one of the treatments we're going to be talking about, which is CAR T. Pat, you're very welcome.
Thank you. My name is Pat Byrne. I'm a cancer survivor. I'm recipient of CAR T therapy. So I'm just going to give you a few minutes about my journey through and to CAR T. So I was diagnosed in early 2021 with high-grade non-Hodgkin's lymphoma, blood cancer. And I was referred to Professor Bacon by my GP. The word from my GP and from Professor Bacon was this is very treatable disease nowadays, and the outcomes are quite good, and we could expect, that I would be in remission after some treatment. So a treatment plan was laid out. I worked through that, trying to get term with all the terminology, CHOP and R-CHOP and ice and rice. So we worked through a 6-month plan, and as we came towards the end of that, it was obvious that despite the best efforts of Professor Bacon and the team, that the chemotherapy was not going to work for me. During that treatment, Professor Bacon had alluded to a new treatment called CAR T, and at one of our consultations, he suggested that I might be a good candidate for CAR T therapy. So with not many options after that, I obviously took the chance to avail of the therapy. So over a number of days, I was briefed by Professor Bacon and the CAR T coordinator, Liz Higgins, all about CAR T. I didn't know anything about it, where we would probably know something about chemotherapy, but CAR T was completely new to me. I hadn't even heard the term. And while I was focused on the chemotherapy, CAR T just didn't come onto my radar. So what it entailed was towards late November 2021, my T cells were harvested. They were sent away to a laboratory, I believe, in France. And in early 2022, I was infused with my modified or engineered T cells. I had a number of meetings with Professor Bacon and the transplant coordinator, the CAR T coordinator, and obviously there are numerous side effects to this, as there are with chemotherapy, but I was well prepared for the side effects. Little did I think that I would receive all of the side effects, but I was prepared. The team had me well briefed. So on the 10th of January 2022, in 11 minutes, I was infused with my engineered T cells. Almost within 12 hours, I started to get some of the side effects. Sepsis being one of the ones that it's not too nice, but brainstorms, neurotoxicity, so I had coordination issues, lots of things like that. So the one thing I can say was that it was an amazing team. There was a whole of hospital approach to my care. I ended up in ICU, but everybody was well briefed and well able to cater for me. So within a number of days, I was released from ICU and started to make progress. And at the end of January 2022, I was released from St. James's, and by March of that year, I was back losing golf balls. By September, I was able to go on a foreign holiday, and by January of 2023, last year, almost 1 year to the day, I was able to go visit my grandkids in Australia, which was something I hadn't thought I would have been able to do the previous year. So for me, CAR-T has given me my life back. Not that I lost it, but there was a year in which, between chemotherapy and the CAR-T therapy, that is missing. But the life I had prior to receiving the CAR-T and the life I have now are exactly the same. I'm on no medication. I have suffered no side effects other than the initial ones as outlined by Professor Bacon and the team. And I'm living my life exactly as it was prior to my diagnosis. So that's just a short synopsis of my journey through CAR-T.
When I asked Pat how's he doing now, he gave the customary Irish response, which was wonderful story and a really great illustration of the potential and power of some of the medicines that we're going to be talking about. Pat was 1 in 10, and we want that to be a much lower number, right? Or a bigger number? Bigger number. I want to get that one right, don't we? Okay, so Pat, thank you very much for that, to sort of give us that light that we're all working towards. We're going to have our first panel now, and it is about immune checkpoints, discoveries to trial and beyond. The first checkpoint antibody was approved by the US FDA. It was ipilimumab, approved in 2011 for the treatment of melanoma. Since then, we've seen nearly a dozen FDA-approved checkpoint inhibitors approved. This esteemed panel of true experts in this area, so we're delighted to have them all. They are Dr. Jim Allison, a Nobel laureate and chair of immunology at MD Anderson's Cancer Center. If you can join me up as a co, that'd be great. Thanks very much, Jim. He is renowned for his pioneering work in T-cell regulation and cancer immunotherapy, covering enhancing immune checkpoint therapies and discovering new targets. He also went straight to the pub when he arrived on the soil of Ireland, which is exactly the correct protocol if you're not from here. Also, Dr. Pam Sharma, who's a professor at MD Anderson, focuses on tumor rejection and resistance in immune checkpoint therapy. Thanks very much, Pam. And finally, Professor Jarushka Naidoo, who's a consultant oncologist at Beaumont Hospital here in Dublin and a professor at RCSI and Johns Hopkins. And she is also an internationally recognized expert in immunotherapy for lung cancer, melanoma, and immune-related toxicity. So, fantastic panel. Before we get to our visitors, Jarushka, would you mind just maybe giving us a sort of an overview? What exactly is a checkpoint inhibitor, and why did people think that this T cell would be was a good target for this sort of idea of using immunotherapy?
All good. I was just going to say, I'm going to defer to, this is like talking about the Bible in the presence of God. So in terms of this, I think I will hand over to Dr. Allison. Dr. Allison.
Okay.
Yeah, well, immune checkpoints are molecules that regulate mostly by limiting the ability of checkpoints to attack their targets. We discovered the first one called CTLA-4 in the early 1990s in mouse experiments, just looking for molecules that were involved in regulating T cells in normal immune responses, and came across this molecule. It occurred to us that one of the problems with cancer is that they do not elicit a quick immune response like viruses do and bacterial do, because they aren't really perceived. They don't have all the signals that those do to elicit the innate immune system, which I assume you'll be hearing about later, which is involved in starting these specific targets, attacks towards bacteria and viruses. But tumors don't do that. They're so close to yourself, they really aren't recognized as being foreign. And so they get to grow a while before the immune system actually gets started. And these checkpoints can kick in on the T cells and stop them from generating the army. The story is you've got about probably 10 to the 10th power, 100 billion or more T cells in your body with different receptors. So you've got a vanishingly small number of any of them. You have to have an army to go attack the cells. So when one sees something on a tumor that's foreign, those cells have to expand to huge numbers. And the job of CTLA-4 is to stop that after a while so that the T cells don't take over your body. And so when we discovered that, I had the idea that maybe we could, by blocking that checkpoint, just let the T cells keep going and take out tumors. And so that was the origin of it. And since that was CTLA-4, since then there have been a number of other molecules that work in more or less similar ways, not exactly, but also limit T cells. And just by taking them out, so we can let the immune system.
So it was almost by chance that you came across this? You were just looking at molecules, or had you always the idea of...
We were looking for molecules that regulated in positive or a negative way.
Okay.
But it wasn't specifically for cancer. It was just to understand how T cells work.
And just for me, because I'm a non-specialist in this area, why don't the T cells attack the cancer? Why is the checkpoint there in the first place?
Well, it's there to limit that proliferation. And you have, like I said, you have these small cells. They start dividing almost as fast as bacteria when they're multiplying. They generate hundreds of thousands of cells within a period of a week or less. And you've got to stop. That's got to be stopped, or they will literally take over your body, as the same way that leukemia would, for example. And these are the molecules that keep them in check and say, no, you've done your job. Stop. Don't hurt things. It's a way of preventing your immune system from hurting you, and that's basically it.
When you recognized this that as a potential therapy, what were the biggest challenges in developing that first drug?
Well, the first one was to find somebody that would help us make one, because at that time, people were convinced that immunotherapy would not work. And so for several years, we went around trying to find a drug company that would help us make the antibodies, and they said immunotherapy will never work. It's been tried for a long time and hasn't worked, so it never will. And finally, we found some people.
Why did people think immunotherapy wasn't going to work?
There'd been a lot of attempts to make vaccines early on and things like that, and use cytokines and things that the immune cells treated, and had sporadic successes, but overall had not worked. And so people were looking for chemotherapy, genetically targeted therapies, things like that, to do it. But I think that the reason some of those things didn't work was not because the ideas were bad, but because we didn't know enough about the molecules that regulated the responses. The basic science wasn't there to understand how to do it in precise enough a way to avoid damage to just having things go awry when you start broadly trying to release a response or too narrowly try to activate one. So.
Can you talk to me a little bit about what it's like to get a drug that actually gets to market and actually does something that didn't seem possible at the time and has had incredible effects on the patients that it treats?
It felt really good. I mean, particularly the first time I met a patient who had benefited from it, it really changed my life as well as other people's too. But no, to do basic science, I'm a scientist and was doing it for science, but I always kept, I wanted to do good with my science, and to have something that finally worked, was icing on the cake, for sure.
Can you maybe just tell me a little bit about the response to immunotherapies, Pam? So tell me a little bit about your work, because sometimes we talk about hot and cold tumors. So when someone gets an immunotherapy, what do they mean by that? How do we differentiate by that? And maybe tell us a little bit about your work.
Yeah, so I'm going to just step back a little bit, just so we are all on the same page about understanding what the immune system is and how it operates inside your body. Because as you're sitting here, all of your immune cells are really circulating in your body as you're sitting here among us today. And that immune system is actually made up of many different types of cells. And these cells are called T cells, B cells, NK cells, macrophages, dendritic cells. These are all the different cell types that make up that immune system. And they're circulating in your body looking for what does not belong there, because they've been well trained on what belongs there. So your immune system has been trained from the very beginning to know you, just individually you, not the person next to you or somebody else, just you. And so when something is there that doesn't belong, it becomes activated. And so that's the response we're talking about in terms of when you get a viral infection or a bacterial infection. That immune response immediately kicks in to say, I need an army, so I will proliferate and make multiple types of myself. These T cells are the soldiers, and these T cell army will then go and get rid of the virus. But you don't want those T cell army to stay forever. You don't need an army circulating all the time. And that's why we have the checkpoints. So those immune checkpoints like CTLA-4, PD-1, they stop the T cell response so that it's not attacking the body all the time. Because imagine if the response keeps going, that's how you end up with autoimmune disease. So you end up with rheumatoid arthritis and lupus and these other things that you don't want. So there's this checks and balances of the immune system. You can turn it on, but you must limit the response. And that's called homeostasis or balance. So it's always in balance, your immune system. It gets going and then it turns off. The issue with cancer is that cancer is so much like your normal self that the immune system is not quite sure when it should start doing something. Cancer doesn't have all the same signals that the bacteria or the virus has. And so you don't get an immune response right away. And even when you get the immune response, the cancer has already had a long time to grow before the immune system recognizes it needs to do something. And so the immune system can get turned off very quickly because, like I said, the checkpoints will come in and say, don't keep going. So you'll kill some cancer cells, but those cancer was growing a long time. So it had a head start to the immune system. And the checkpoints kicked in to stop the immune response before the cancer was completely eliminated. What Jim's drug does is it then takes the brakes off the checkpoints. It says, those checkpoints, we don't want you around anymore. CTLA-4 needs to go away, PD-1 needs to go in. So the antibody he created stops the checkpoints from working. And so even though the cancer cell had this lead time, the T cells can now catch up. And now when the T cells catch up with the immune checkpoint therapy, the cancers can be eliminated completely. And this was really a game changer for the entire field of oncology, because we never had a setting where patients who had tumors throughout their body, liver, lung, lesions throughout in different places of the body, lymph nodes, and you can now see all that disease go away in a subset of patients. And so this gets us to the idea of why only a subset of patients. What's happening with hot and cold tumors? So melanoma and lung cancer, for example, those tumors do very well. About 20 to 40% of those patients will respond very well to immune checkpoint therapy. But cancers like prostate cancer or glioblastoma don't respond as well to these immunotherapy agents. And that's what defines the cold versus hot tumors. Hot tumors are the ones where the immune checkpoint therapy is working. Cold tumors, they're not working. And so why is that? And so it gets us back to what we heard initially. Some tumors just have lots more T cells and immune cells within the tumor. And so because they have lots of T cells already present, when you block the checkpoints, those T cells can keep doing their job. And in some tumors just don't have the T cells. Even if you give a drug that blocks the checkpoints, well, there's no T cells to block the checkpoints on. So then you're not getting the responses. So how do we get the T cells into the tumors becomes the next question. And for that, we need combination therapies. And we need rational combination therapies, meaning that you're going to give a therapy that can maybe attack the tumor. Maybe it's chemotherapy, maybe it's radiation therapy, maybe it's another targeted agent therapy. It will attack the tumor, allow for some of the tumor cells to die, and to start that immune response so the immune system can go, hey, there's some death happening, which means I need to go and do something. And then you can give that immune checkpoint agent to take away the checkpoints and get the T cells to even get to the point of rejecting it. And I think there are lots of different combinations to think about that people are trying to test and see which ones will work in glioblastoma or pancreatic cancer. And these are still questions that we're researching in a lot of different ways. The key to all of this is really understanding the human immune response. What Jim said was that the basic science and learning about the immune system in mice and how T cells work in mice led him to his discovery. And that's called the basic science, because we're really looking at it in, in those cell types or in the murine models. But you have to then get it to patients, and that's the first clinical trials. When I wrote the first clinical trial for immune checkpoint therapy, that was years ago. We didn't understand how it would work in humans. Actually, the mice never had toxicities, for example. And humans get toxicities from these agents. So it's
those
very
toxicities
different.
come from, where the toxicities come from, from overactivity
From
overactivity of the immune system? Yeah.
Exactly. The same immune system that's getting rid of the cancer is now an immune system that's going to attack maybe your colon or your lungs, and so it's going to cause damage to normal tissues. And that's what we can see in terms of the toxicities. So what we tried to do is get a combination that can give us efficacy but minimize the toxicities. And I think the research now is about understanding what happens in the humans. So the basic science was very important to understand T cells, but now that we're in the clinic, we have maybe 3,000, 4,000, 5,000 clinical trials ongoing, and some of those are not working, but we need to take the samples from the patients, get the tumor biopsies and the blood samples to understand what's happening. And what we've been finding is that it's not always an issue of just the T cells. Those other cells I told you about, the macrophages, the B cells, the dendritic cells, they're also important. So if a tumor has lots of macrophages and not enough T cells, then we have to learn how to target the macrophages. But in order to target the macrophages, we have to go back to the basic science, because the basic science Jim gave us was on T cells. We still don't have enough research about the basic science to understand macrophages in enough depth that we can come back. And even macrophage is a basket term. There are many different types of cells that make up this macrophage subset. Yeah. So I think it's back and forth between the lab and the clinic for us to really make progress. And so that's where the research that I've been doing is how we go between the patients getting the samples, understanding from the patients in the lab, designing experiments in the lab to understand these other cell types, come up with a new target, and then come back to the clinic with a combination strategy.
You explained that really well. Thank you. Thank you. Jarushka, your work is in tumor rejection and resistance to this immune checkpoint therapy, which is exactly what we're talking about here. Can you tell me a little bit about the Irish context and the decisions you have to make?
Yeah, so following that is tricky, but so I'm a clinical trialist and a lung cancer specialist, and I went and did my fellowship at Memorial Sloan Kettering, where both Dr. Sharma and Dr. Allison were for a time before they went to Texas. And at that time, actually, interestingly, as Dr. Sharma mentions, now we appreciate lung cancer and melanoma are hot tumors or tumors that respond to immunotherapy, but back then, it was almost heretical to think that lung cancer would respond to immunotherapy. I remember being a fellow here in Ireland. I trained in Ireland, even though clearly unusual Irish. I'm South African, and I went to college here in Trinity and then won a scholarship from the Irish government to go to Memorial. And at the time, I remember telling one of my consultants, I think I want to do lung cancer. I really like people who are sick. I want to fix people who are sick. And I saw this phase 1 clinical trial published in the New England by the first author who would end up being my mentor. Her name is Julie Bramer. And there were 3 patients with lung cancer who responded. And they said, that's nothing. Lung cancer is going nowhere. Chemotherapy, you're done. And I was like, well, that must be something. And I think I will do immunotherapy for lung cancer. And lo and behold, we learned a lot from those 3 patients. So lung cancer is not all created equal. There are some lung cancers that particularly happen in never smokers that don't have some of the classic mutations or alterations that allow the immune system to recognize it as foreign. And therefore, some patients with lung cancer, unfortunately, this immunotherapy is not the right choice of treatment for them. But for other types of lung cancer, perhaps more typical lung cancers that may be associated with smoking or other environmental factors, they have lots of what's called non-synonymous mutations, meaning genetic alterations that happened as a result of smoking, the same way there are lots of mutations that happen in melanoma as a result of sunlight. So this means that the immune system recognizes these tumor cells as foreign, and that subset of patients with lung cancer appear to respond to immunotherapy. However, the topic that Jonathan is bringing up, not every patient responds to immunotherapy, or they have a response, but for some reason the immune system outsmarts that response, so there are other pathways and mechanisms that come into play that mean the cancer loses its response. And this is a term called resistance to immunotherapy. So many of us, like Dr. Sharma is explaining in the clinic, you might see a consultant if you're a patient with who's being treated with immunotherapy or any kind of treatment, and we might want to talk to you about signing up to what's called a translational trial, where we will take some samples, blood samples, even now stool samples, to understand some environmental factors that may be playing into why a person's tumor may respond or may not respond or may lose its response. And many of these types of trials, while they don't necessarily bring a treatment to a patient, they give us the building blocks that allow us to create the treatments of the future, to outsmart resistance to immunotherapy. And another area that I'm particularly interested in is toxicity, to understand why some patients develop toxicities versus others, and is there a way to write clinical trials to try to make immunotherapy safer by adding some kind of a modulation to reduce the risk of toxicity. So really, this is a story of incremental gains, that sometimes we have to question some fundamental dogmas, the way Dr. Allison, well, immunotherapy doesn't work. No, it can, just for the right patients and with the right tool. Similarly, well, immunotherapy doesn't work for lung cancer. No, it can, just for the right patient at the right time. So I think we have to question some of these, and understand as patients and the public and all of the people who are studying this, that treatment for cancer is not a one-size-fits-all. We just have to find the fit that might fit you.
You make it sound really complicated, Jarushka.
Really?
No, the science, no, you explained it really well, but the science, it sounds like it's very, very complicated to figure out all of these different aspects. And so for Jim, for you, since finding that first checkpoint inhibitor mechanism and getting a drug that works, are you looking at different targets? Are you working at refining that particular approach, or what is your research at now?
Yes, I'm working with Dr. Sharma, and we are looking at a number of things. My own lab is looking at additional checkpoints, which differ a little bit from their original ones, and some of these can be found on the myeloid cells that she mentioned as well, and can inhibit cells in a slightly different manner. But if you can take those out too, it's got the possibility of, adding another checkpoint blocker to remove an additional one and make it go further. The example now, I guess, is not exactly that, but with two different checkpoints that are out there now with melanoma, the ipilimumab could cure, meaning 10-year plus 20-year survival of patients after a single round of treatment. We now can get up to about 55% of melanomas, which was in 2011 when ipilimumab was approved. Before that, the median survival was 7 months after diagnosis, and virtually no one was alive at 5 years. Now, better than 50% of people are alive at 5 years that receive a combination of two antibodies. And recently, there was a trial combining that with another surgical technique called neoadjuvant therapy, where the response rate, it's a trial that's still going, but the response rate was almost 80%. 3 years. And so, and we think if we could add, the myeloid cell component of this, we can get that up. So I think the future is identifying these additional ones, and it may be 3, 4, 5 different things, as long as they don't add toxicity, by bringing the science to bear and checking them out. Because what we do know now is when you perturb one, many more pop up. The immune system adapts. It's always adapting to what you do, and it's trying to stop that from running. And so if we can just find other ways, because when you remove these therapies, the system pretty much comes back to normal. But T cells, at least, there's a population of them that become memory cells and are there for the rest of your life. So if the tumor comes back, you're protected again. So
I don't know how we're going to get this through in the required time, because this is brilliant stuff. Pam, before, I want a quick question for yourself and then a quick one for Jarushka. So quite a short, if you can. Tell me about your current clinical trials, doing one with bladder cancer. Where are you at?
The immunotherapy platform at MD Anderson is something that I set up when I started at MD Anderson in 2004. It was first in just GU tumors, now it's institution-wide. The key there is that there are going to be pathways that can be translated across tumor types. For example, CTLA-4 works, blocking CTLA-4, blocking PD-1 works in bladder cancer, lung cancer, head and neck cancers. So there are pathways that it doesn't matter about the tumor type because we're not targeting the tumor, we're targeting the immune system. And then there are certain pathways that we're going to have to think about that it's specific for prostate cancer or specific for pancreatic cancer. And so designing a clinical trial is going to be important to think about that. Also, it's going to be specific for, does the patient have lung metastases versus liver metastases? That may also be having to design a trial in a different way. The other thing that I think is important is that a lot of times when we've designed clinical trials, we design them for chemotherapy. So we have this habit of doing everything based on what we've done in the past for chemotherapy. And that means using maximum tolerated doses. We go as high as we can in the doses for chemotherapy. And it means combining in a certain way that maybe is not right for immunotherapy, because immunotherapy doesn't require maximum tolerated doses. So I think we're trying to now step back and think about how to do smart clinical trial designs for immunotherapies. And that's where the work is going at this point, especially in the neoadjuvant or earlier stage of disease, because we think that's the place to get the most benefit from immunotherapy.
Great. One thing we haven't really talked about is, Jarushka, what is life like on these therapies long term? And I'm just wondering, how do you manage those patients who are on immunotherapies for some time? What is that like, and what are the things you need to think about?
Yeah, that's a really good question. And to be honest, it's a great problem to have, that people are surviving. So now we're in a new era where people are surviving, but obviously we want them to thrive as well as survive. So I think we see lots of different types of patients in this situation. Obviously, the person we're clinking champagne with is the person who has no side effects whatsoever. Their tumor has remained stable, or maybe in some instances completely shrunk. And we only see them, maybe every couple of months. We do a scan, we shake hands, and that's all we need to do. But on the other end of the scale, and certainly I have quite a few patients like this, we have patients in whom they may develop side effects of immunotherapy that might last for a prolonged period of time. And then we have to become specialists in other types of organs, not just the cancer. So I've become a little rheumatologist, a little sort of specialist in joints now. Oh, how are your joints today? And learning all about the different types of immune treatments that we will give to try to minimize a patient's joint pain, for example. So we do have patients who are affected by the long-term side effects of some of these treatments. Thankfully, in general, this is a rare circumstance, a very rare circumstance, but it can mean that it affects the day-to-day quality of life of a patient. So now we're seeing new information generating, an immunotherapy survivorship plan. What are some of the things that we need to tell patients ahead of time, and the ways we need to monitor them, like monitoring their thyroid function and all of the other organs that may be affected by immunotherapy. So certainly when we manage these patients long-term, we're in a new era, and also this raises new questions. So certainly we're taking a lot of blood samples and other samples from these patients to understand what's the secret source of a long-term responder, but also what are those elements that make patients have long-term toxicity, and how could we potentially mitigate them from the start?
Jim, I can't let a Nobel laureate leave the stage without asking, when did you get the phone call, and what were you doing at the time, and what was that like? I know you've been asked that 1 billion times, but for this audience, when you get that phone call, what is it like?
Well, it was a little strange because I got the phone call from my son.
Really?
Because, well, he got up. I didn't wake up in the morning expecting to get a phone call, early or anything. But he got up early and was watching the press conference early, Swedish time, and he actually called me and said, "Dad, I just saw you on TV. They said you won the Nobel Prize." They had my office number. They've been calling my office number, and we were in New York because Dr. Sharma was receiving a prize. I was with her. She was receiving a prize for immunotherapy from a society that we belonged to there, and I was her guest, accompanying her, and so we thought maybe it was somebody, they were calling about her eventually. But anyway, my son called. We began to have a celebration, and finally somebody got my cell phone number to them, and then they called me, and my son got the chance to film it on his iPhone.
Oh, nice! Because I've interviewed
so
a lot.
It's a little different than most people's experience.
Yeah. Well, I've interviewed a lot of people. No one has ever not found out from Sweden, as far as so that's hearing from your son. That's lovely. A very winning partnership, clearly. I would love to spend more time talking with you, but thank you so much for joining us. Please give a round of applause for our panelists, please. Jim, Pam, and Jarushka. Okay. One of the things in my job is I have to get close to the time that we agree that we would do, so it's sometimes it's brutal. So thank you so much for our panel. The focus of this next one is on CAR-T, which I'm sure you may have heard of. CAR-T was first approved by the FDA in 2017 for the treatment of pediatric and young adult acute lymphoblastic leukemia. We heard earlier from Pat, who's the second Irish patient to get CAR-T in 2022. Delighted to welcome Professor Larry Bacon. He's a clinical hematologist at St. James's, specialises in lymphoma, acute lymphoblastic leukemia, bone marrow transplant, and so on. Owen Smith, please applaud them as they walk on, so we're trying to get through. Owen Smith, Professor of Oncology and Hematology at Trinity College Dublin, visiting Professor at UCD, NCCP National Clinical Lead for Child, Adolescent and Young Adult Cancers, and consultant pediatric hematologist at CHI, Children's Health Ireland. Dr. Bernie Fox is an expert in tumour immunology and immune profiling. He spent 30 years advancing cancer immunotherapy and currently leads the Laboratory of Molecular and Tumour Immunology at the Earl A. Childs Research Institute. And finally, Dr. Nicki Panoskaltsis is a hematology oncologist at St. James's Hospital and Professor of Personalised Therapeutics at Trinity. Thank you all for joining us. Larry, if you don't mind, would you start off and explain to the audience what CAR-T is and how does it work?
Okay, so CAR-T cell therapy is one of the more advanced novel immunotherapies that are available. We've heard that the T cells, in oncology cancer, is trying to switch them off. I guess what CAR-T cell therapy is actually going to start is trying to turn them on. So for some patients, like Pat, who has spoken to us, his tumor didn't go away after 2 separate lines of chemotherapy. So there was, probably something wrong with his T cells. That at least is where the CAR-T cell therapy came from. So what happens is that you take the patient's own T cells, you take them off in a process called pheresis, and they get away and they get re-engineered. And not just re-engineered, but you also get to make much more of them, okay? So those cells are engineered to I suppose, find that cancer and find it quickly and eliminate the cancer. So in a way, going back to Jim's thinking about an army, what you've developed is a whole new army, but you've given them better ammunition, okay? So when they're reinfused into the body, they're there and they start to grow, so they're living drugs. That's unusual, okay? So the T cells get in, they start to grow and multiply and get up to billions and billions of cells. That's where the side effects come from, is they're overwhelming that PD-1 blockade. But not only are they overwhelming, but now they know what they're looking for. And I think we had talked that maybe they can't find the cancer, they're hot or they're cold. These guys are targeted, so they know where they're going and what they're looking for. And they're made from the patient's own cells. You don't have to worry too much about things we worry about in bone marrow transplantation. We won't get into that right now, keep it simple. But these cells do have some toxicities, but they're a really good targeted cell for people who have not responded well to chemotherapy.
Right, I want to get into, actually I had a good chat with Nicki about the engineering, which I'll talk to you about that in a bit, but can you talk to me a bit about the CAR-T program at St. James's, how it's evolved and how it's doing?
So the CAR-T cell therapy started actually with myself and Owen had met with the companies and the CEOs of the hospitals, with HSE, our managers, if you like, to set up this program. We met, I think, in 2017 when we started talking about this first. It took about 3 years to develop that program. Most of the time was spent getting funding for the drugs. I have to be honest about that, and that was a problem. But we eventually got it open. So as Pat said, he was the second patient just in January 22 to get up and running. And in our first year, we got through 20 patients. Our second year, gone through 30, and this year we're heading up on somewhere between 40 and 50 patients, we guess, by the end. So already over 100, well, hopefully up to 100 patients by the end of this year will have benefited from CAR-T cell therapy. So, it's what we planned for and we'd hoped that we would get to. We would like to do more. I mean, right now we've only 4 patients waiting on infusion, so we have done pretty much everybody who needs it for now. We're limited in two things. One is we don't have enough capacity to get people in as quick as we'd like to. But the other thing we're lacking is funding for the drugs. So we have two great drugs, but we have them for a limited number of indications that they're available for, and we can't get the newer drugs in. So because they're a drug, they have to come through a funding process. So again, they're expensive. Nobody's saying that these are cheap. They are very expensive drugs. We need to get, but getting them funded is what's holding up the program, as well as having beds. So I mean, if somebody turned around to me tomorrow and said you can have all the drugs and all the people you like, I don't know where I'm going to put them, but I don't think anybody's going to tell us that. But for now, we have a very successful program. We have, as I said, up to over 80 people already reinfused. Thankfully, we've had no mortalities. And we've had toxicities, and as Pat has said, we have a lot of people who've gone up to ICU, but have all come back. And again, that's when we set up the program well when we started. So we were up and running before we got running, which is unusual, I guess. Often as physicians, we jump in and then sort of worry about the toxicities later. But this way so we were late getting to the program. We were 22. The first, had started in 17. But you know what? We learned from the other centres who were doing these, so we were well versed in
Sometimes it benefits to be a little bit, but not always.
Sometimes it's, but don't tell the HSE that.
Yeah. Just on the funding side, does the cost mean that it's always a last ditch treatment, or would you ideally like to be giving CAR T much sooner on?
Yeah, that's a good question, and in truth, the reason that it is third line is because it's an expensive therapy.
Right.
So already trials have shown that if you use it in some of these drugs in second line therapy, that you could do even better. We don't have that funding for that therapy yet. And we've just been involved recently in a clinical trial for multiple myeloma, somebody mentioned to earlier, and we had access to that CARTITUDE-5, that patients getting the CAR T-cell therapy up front. So
wow.
that was their first, part of their first line, first term of defense. So can't wait to see what the results of that are going to show. It'll take a long time just because myeloma is a chronic disease, so it'll take a long time before we know how beneficial that was. But we've got that through clinical trial.
Owen, when it comes to pediatric patients, do you have to think more about CAR T because they're kids?
Yes, in a way, in terms of toxicity, because what we're doing now with children, with the commonest cancer in children, acute lymphoblastic leukemia, we're curing in excess of 90% of those first time around with chemotherapy. But that cure comes at a cost, and the cost are long-term side effects. So when CAR T, as we've heard so far from Larry, I mean, the indications are usually refractory or relapse disease. So the horse is bolted at this stage. And what we'd really like to do now, in combination with the other antibody-directed immunotherapies like blinatumomab and inotuzumab, is to bring CAR up front in de novo patients to offset some of this long-term toxicity.
What does that look like?
Long-term toxicity looks dreadful because when you look at some of these survivors of combination polychemotherapy, 20 years, 40 years down the road, they've accumulated 6 to 12 different chronic diseases. Heart disease, brain disease, neurological disease, it's horrendous. And we've just heard earlier on about survivorship. People in adult medical oncology now are looking at survivors now, where in the past they didn't. We've been looking at this for the last 50 years prospectively, and it's a dreadful, dreadful scenario. Right. So we need to be clever about, and we've already heard from this, is how to design clinical trials going forward to incorporate these particular novel immunotherapies and try to get rid of the nasty genotoxic chemotherapy and bring them forward. And we think we have ways of doing that now.
So what's your approach when you're talking about complicated cases? How do you go about figuring out the approach, and what treatments are available to you?
So it's a changing landscape rapidly at the moment in terms of genomic sequencing. So we've got these exponential technologies that we're hearing about, how to gene manipulate certain genes within your genome, but also using sort of machine learning to help us deliver that as well. So what we're doing at the moment is we're laying the landscape bare from a genetic point of view so we can pick out the bad actors up front and target their particular leukemias or their lymphomas in children, and then move that on. But at the same time, we're looking at the germline genome to look at those variants in your genome. So why does one child develop osteonecrosis of hip and the next child doesn't? We need to be really clever about picking them out at diagnosis and saying, not a lot of steroids for this kid, more immunotherapy for this kid, etc. The cost will ultimately come down, but what we're doing there, Jonathan, is we're redefining cure. So cure of cancer is really cancer-free with no toxicities.
Yeah.
And up to now, we haven't been doing that. But these technologies will allow us to advance that.
Yeah, and you talk about genome, you're talking about the genome of the cancer?
So we're talking about both. So when we talk about the cancer genome, that's the somatic genome. That's where all of those nasties are that's driving the cancer. But we need to look at the germline, what you've inherited from your mom and dad, to see are there predisposition. And we know now that 1 in 6 children who come to us in the Children's Hospital with cancer have a genetic predisposition that's causing that cancer.
Right.
We're also on the other side, looking at the genome, what genes there are going to predict for certain patients to develop toxicities. And then we can intervene by stopping those toxicities.
But when we just from my own understanding, when we take the T cells out and we engineer them, we're not talking about genetically engineering them before they come back in. It's adding proteins, it's adding receptors onto those.
So we're just re-engineering the receptor, the T cell receptor, to have basically an antibody at the end of it that recognizes specific targets.
Yeah, you're
and
giving it a mug shot essentially.
Exactly. So now we're seeing that 50% of the patients who we get a very good response in children with relapsed ALL, 50% of those relapse further after a CAR T. So the real insight will come, can we predict those patients who we know are going to relapse after a CAR, don't put them into a CAR, put them into a bus, and give them an allograft. Yeah, so we switch things. We'll save money that way as well.
I want to talk a little bit about how we engineer these cells. So, Nicki, we were talking about when we take the cells out, that we need to do more than just a one-size-fits-all. Can you explain to me what we do with the approach to these CAR T technologies?
So currently, as Larry was saying, we take cells out from a patient using an apheresis machine. That's a bit like a massive machine that filters the blood, and a patient will go on the machine for about 3 hours a pop. The cells will be filtered up that we want, the white cells into a bag, and then everything else is put back into the patient. And that takes about 3 hours, so it's constantly filtered. Those white cells are then taken, and as we heard previously, there are different types of white cells in the system, in the body. And so those are then taken off to a lab and separated out for the lymphocytes or selected out in some way for those cells, in particular the T cells that we heard about before, that can be activated and multiplied to make more of these Trojan horses, per se, or more of the army cells that we need. And as they are activated, they are engineered to express on their surface of these cells a receptor that can target a protein on a cancer cell, such as CD19 like we have in our lymphoma cells, in order to create more of a bullet type of targeted therapy for these lymphoma cells. And they're multiplied in this way in culture that can currently take several weeks, although right now people are trying to limit that time frame so that we can get the treatment back to the patient faster, because the quicker we can get that treatment into a patient, the less time they need to wait for and get other treatments to keep the cancer at bay. And so when we get it to multiply, those cells change. Not only are they changed because they're engineered with a protein to target that cancer, but they also change in terms of the types of T cells. So there's not just one type of T cell, there are different types of T cells. Some of them are more active at fighting off cancers, and some of them are less active at fighting off cancers. That's a very simplistic view, but some of them are more metabolically active to target into the tumor, and some of them don't last very long. What we want is a longer lasting T cell that can target the tumor cells, that can be viable and metabolically active to do the job that we need when they get there. Because these are living drugs. It's not like the antibody therapies that you hear about, the checkpoint inhibitors, or targeted therapies that you have for a gene, for example. They're living drugs. So as they're being made, they change. And even after they're infused into a patient, they change. And so we want to get a drug, a living drug, that is going into the patient and can target that tumor and stay there and do the job that it needs in the patient, but not to expand too much, because then you're going to get problems like the adverse events, the adverse effects, and the toxicities we've been talking about.
Yeah.
So you want to get the maximum efficacy, the maximum bang for the buck, literally, and reduce the toxicity as much as possible.
Are there treatment options we can use right now to reduce the toxicities that we're using?
So, and Larry can probably speak more to this than me, but right now what we do is we give one dose and one size fits all, okay? And people are individuals. We're different. We're not all the same. We can't use one drug for everybody and we're going to know what the outcome is. We hope that the outcome is the best possible. But right now, we have a one drug fits all, and we're using it at one dose. There are strategies where you can microdose, so you can limit the doses and increment them, and that's reducing toxicities. That's not something that we can do, unfortunately, with the cells that are manufactured and then bought.
Right.
Because it's one dose, and that's the way that the cells need to be given to the patient. The drug needs to be given to the patient because it's a drug. It's manufactured.
Yeah.
It is. It's sold as a drug.
Yeah.
And you give one dose. But there are other ways that people are doing in very exciting and novel research that you can change the types of cells as you're manufacturing them, or putting different cell types together, or different engineered proteins in the cells in order to shut them off when they need to be shut off, or limit the toxicity.
Can you tell me about your work trying to understand how best to personalize this kind of approach? Because you're doing a bit of work trying to getting these sort of collaborations together to understand how we can best personalize these cells.
So it's not just me, it's a consortium. Larry and Owen and A. Dean are part of this consortium in Ireland. It's called IMPACT, the Irish Medicine's Personalized Advanced Cellular Therapeutics. And we're trying to personalize the cell therapies in order to improve our efficacy and reduce the toxicity. And how are we doing this? At 7 universities, 5 hospitals nationally, and 4 organizations. And it's an all-Ireland consortium to try to get all of the pockets of expertise we have in Ireland, which actually is fantastic expertise in immunology in particular. You need good immunologists for this. In manufacturing, we have a very strong biomanufacturing sector. In informatics, which you need to get these, all of the data sets that we're putting together for patients, genomic, cellular, the phenotypes of the patients, the phenotype of the disease, the phenotype of the cells that we want to put in, these living drugs, and how to do that better. And really putting all of these great things that we've got in Ireland together to get better drugs to patients faster, manufactured with high quality, and with reducing the toxicity. And that's the way that we want to try to personalize these therapies, because, you know, Ireland is very late coming to the game of cell therapy, unfortunately. And even now, we've only got 2 of the drugs available to others in the world that our patients could have access to. So for a patient with multiple myeloma, unfortunately, they don't have access to that CAR T right now in Ireland. And why? We are a wealthy country. We're one of the wealthiest countries in Europe. Why do we not have therapies? And I'm going to put a call to action
I have been
here.
asking
I'm going to interrupt you.
Over and over and over.
We need these drugs for our patients. Our patients are losing out. And you need to talk to your politicians. This is important. And it's not just cell therapy.
Yeah, and every time I ask a different stakeholder in this discussion, they all say, we all need these drugs. And I'm like, well, where's the problem here? It absolutely is something we need to address. Byrne, hematology, we look at these sort of approaches that work really well when it comes to solid tumours. There's other challenges. Can you talk to us a little bit about that and your research?
Yeah, I think.
Or your work?
Yeah. So yeah, there are lots of challenges, and I think they've been alluded to before, and that is that these solid tumors are larger typically. They've got lots of suppressive mechanisms that are active, and Jim alluded to a couple of the checkpoints, but there are so many more, and there are ones that we don't know about yet, and he's also worked, Dr. Allison's working on identifying some of those new ones. But I think that's one of the big problems. So that one of the issues that we've been working on, or been talking with other groups that have got T-cell therapies, not just CAR therapies, but our own group has T-cell receptor therapies for pancreatic and other mutation-based therapies. And that is if the T-cells go in and they go to the tumor site, then you kill off those cells because they get inhibited. The cells in the periphery aren't getting activated because they're not seeing antigens. The idea of adding vaccines as well as with the adoptive immunotherapy, I think, is one of the areas. The other areas to look at, the other inhibitors. Orla mentioned FAST. So the guy next door to me in the office, Dr. Matthew Taylor, has got a new antibody that is a much higher affinity and is looking at starting a clinical trial next year with some of the friends actually that have been helping, that helped Jim develop anti-CTLA-4, Niels Lundberg and Alan Corman, to get this antibody into a clinical trial, and I think it'll start early next year. So there's lots of other agents that are coming down the pike that we're really excited about.
I think it was you, Nicki, you were saying, is it right that the hard tumors, Byrne, maybe you won't answer, but you were saying to me that they're harder to penetrate. Is that part of the reason why they're so resistant?
Well, I think, clearly some tumors you can penetrate. I mean, we see that's part of the monitoring question, right? But I think some of the tumors have got more suppressive influences. You look at melanoma, right? Melanoma is one of those diseases where 40 years ago this December, that Linda Taylor was treated with high-dose IL-2 at the surgery branch when I was a fellow, and she's still complete remission, disease-free, with just IL-2, right? And what was it about her? We still don't know why she was cured. We don't know so many of the mechanisms we don't understand. But she was cured without toxicity. I mean, I get the whole idea with this. She's, so I just think there's so much we don't understand, and that's where the basic science comes in. And then it comes to another question about how do you translate that? Where do you find the resources to do that?
And what do you see as being the biggest challenge in developing a better understanding of why hard tumors are so resistant to?
Well, it's more. I think it's more than just CAR T. I mean, so many parts of cancers, right, that we don't know about. So apologies, I'm not quite the expert in CAR Ts, but thinking more broadly, it just came from about a month and a half ago. The Society for Immunotherapy of Cancer had this strategic retreat, and one of the questions there had to do with exactly this, right? What is it about immunotherapy? Why? What do we have to do to get things to work better? And I think one of the questions that came up, you've got to be monitoring people. You've got to be monitoring them, I think, real time. And there were major pharma partners in the room during this retreat, and we brought up the idea of doing serial monitoring. You've got to stick a needle in a tumor and take biopsies. And they said, well, that didn't work. Well, why didn't it work? Well, they took it, they put it in formalin, they didn't study it right away. And they're probably, if the trial didn't work, they're never going to go back. But if you're taking a drug and you're turning on the immune system, and it's going and it's going and it's starting to make the tumor go away, but some new regulator pops up because you've blocked PD-1 and maybe you've blocked CTLA-4, unless you're taking a biopsy and using the kind of technologies that we have today, where you're taking living cells out and putting them through single-cell sequencing, which is a way you can look at every single cell and all the genes on in those cells, you're not going to know what the other inhibitors are. And we've done that, and we found that regulators that weren't up at the start are now going up, and we presented some of this data at the ACR meeting in March. And I think that's in a trial where now we're up to about a 55% response rate in head and neck cancer, which has the label for that as 16%. And we're seeing in the patients that actually are not responding, they've got T cells in there, but LAG-3 is screaming on the cells that are the 39103. These are a certain subpopulation of cells that are tumor reactive. So that's what I think you've got to do to make all these patients respond. I think probably the same things for CAR T. You're going to engineer certain molecules out of it, but what other molecules are getting upregulated, and not just in the CAR T, what is in the myeloid component in the tumor that's there. So those are kind of the challenges.
Owen?
Yeah, just on that, so it's really important to remember that treating a 70-year-old is different than treating a 7-year-old. And it was alluded to by Pam earlier on as well. When we give these immunotherapies, we need to really survey those patients as they develop into young adults and going forward into older adults, because we don't know what the long-term dysregulation of the immune system are going to be, especially in the children, the young children, and we really need to be upfront on that. And the second point I would just make, we've spoken about toxicity here a lot. The biggest single toxicity of CAR T at the moment is finance. It's not the stuff that puts you into ICU. So we need to get around this. And as Nicki alluded to, the whole concept of impact is to bring academia in with healthcare as well as industry and pharma, and to develop academic CAR T-cell therapies.
And to make them more efficacious and more affordable for our patients.
Exactly.
Yeah.
And I'll just note on that note, there's a colleague of mine, Michael Nishimura, who's at Orla. They developed their own CAR T. They had a program. And those kinds of reagents should be made available, right, for academic centers.
I wanted to ask one question to you about cancer vaccines, because there's some exciting stuff working in there, right? Some exciting developments there.
Yeah, no, I think there is. And if you go back, Dr. Allison, back I think in 1999, had a JEM paper, a Journal of Experimental Medicine paper, where he showed that, you know, a vaccine, CTLA-4, in a tough model to treat B16 melanoma model, where anti-CTLA-4 by itself didn't have much of an effect, and vaccine alone had not much effect, if any, but the combination had a significant effect. And that's the idea that, you know, people say immunotherapy doesn't work. Well, what immunotherapy doesn't work? PD-1 doesn't work, or vaccines don't work. But if you understood the biology and you knew, use the car analogy, if your car is racing to hit the tumor and you take the brakes off, it works better. But if the car is parked in the garage and isn't even turned on, taking the brakes off doesn't help. So the idea of combining things that turn the car on and steer it towards the cancer, which would be a cancer vaccine, something that gives it the gas, which are the TLR agonists and other things that are there, or other antibodies, and then taking the brakes off. That's the kind of combination immunotherapy. Kind of our vision, my colleagues at the Institute, is what are we going to do for combination immunotherapy? And the only way you can develop it is if you intensively monitor patients on trials to understand why they didn't respond. If you just put those samples in a freezer and put them away and you don't go back and look at them, you're never going to learn anything. And the only way you're going to do that is, I want to shout out to this institute, right, this Breakthrough Cancer Research. Philanthropy plays such a critical role in getting these new things forward and in getting the monitoring and the clinical trials. If you don't have that, I think you're going to die. It plays a critical role, and so hats off to. Yeah.
We do philanthropy really badly in this country. Okay.
Yeah. I wanted to ask about the clinic, just to finish this off. In your work, do you look at these combination trials that are happening all around the world? And it seems to me there's a lot of different trials going on, looking at different combinations for treating the sort of patients that you see in your clinic. Like, are you watching those and going, oh, this might work, or that might work, or do you wait until you get really solid results? And
no, we don't wait. We watch, but we don't just watch. We participate. So we were involved in the first blinatumomab trials in the Tower study. So we were, back in 2014, we were involved in that, as that was one of the bigger agents really powerful agent for acute lymphoblastic leukemia. We're involved in clinical trials. We've been very active clinical research.
No, for sure, but there's so many out there. I'm just wondering, how do you know what the right path is for a patient if you see something that might be exciting? Or
So as soon as we see something that's exciting, we go looking for it and try and get them to come. We don't do well in getting big trials in Ireland just yet. I know Jarushka does a lot of work and trying to get them in. We're constantly trying to get them in. I think we've behaved badly in the past, if I can say that, with clinical trials, where people overestimated how many people they're going to recruit. So when I go to a company now, they say, well, last time you said you got 20 people, and they never turned up. But we're improving on that. So we're now getting more and more trials. And I think Owen is dying to talk about how many of the children are on trials in his hospital. but in the adult setting, we are not just watching and waiting. We're actively looking to be involved in clinical trials, and I think it's a big way forward for us. We've done really well in our Caritude 5 studies and stuff. So hopefully with that, more and more companies will come.
Great.
So the thing, as Larry has alluded to, in children, we try and put as many children onto clinical trials because we're small in numbers compared to adult cancers. We have to go pan-global or pan-European. So the 2 trials that we have open at the moment for the commonest one, lymphoblastic leukemia, for the older children over than 1 is an altogether protocol which is across 15 countries across the European Union. It recruits something like, it's the biggest ALL trial in the world. And the second one for the infants who have got a really nasty high-risk leukemia, we have Interfund 21 open now, which is pan-global. We take in South America, across Europe, and some of the Canadian states as well. Yeah.
the importance, if I can interject.
Yeah.
The importance of what we're talking about here is really for people to participate in trials, and we're trying to get the earlier stage trials into Ireland because that enables us to have access to novel therapies quicker, the training that's involved, and actually a chance for patients to have access to a drug that may not otherwise be available. So it gives us that visibility to do that and the ability for patients to really participate in changing things for the future, but also potentially for yourselves. So it's the philanthropy, definitely, to support the research that's really important, but also the participation in the research, and that's how we're going to achieve the personalization, is through the phenotyping of patients going through treatment. So what was discussed before, looking at what's going on in the patients in real time, looking at what's going on in the tumor, and also personalizing these combination therapies, because not one size fits all.
Yeah.
Everybody's an individual.
Brilliant. Thank you so much to our panel. Owen, Byrne, Nicki, and Larry. We're going to move on to our final panel, and then we will take questions. So you can ask questions of all of the speakers who have spoken, but our last panel of this evening is going to focus on the next generation of immunotherapies. Please welcome Dr. Kristin Anderson. She's assistant professor at the University of Washington. She researches engineering T cells to overcome solid tumor obstacles after her own battle with aggressive breast cancer. Alberto Mantovani, he is a Emeritus Professor at Humanitas University and Scientific Director Instituto Clínico Humanitas. He focuses on innate immunity, inflammation, and the role of tumor-associated macrophages. Dr. Jennifer Guerriero, she's an immunologist at Brigham and Women's Hospital. She directs research on modulating tumor-associated macrophages. And Professor Thomas Marron, who's the Director of Early Phase Clinical Trials at the Tisch Cancer Institute and Associate Professor of Mount Sinai, specializing in developing cancer immunotherapies. Jen, if we could start with you, if that's all right. We talked about immune checkpoints and CAR-T in the last two panels. Can you explain that there are other approaches being explored and what they are, please?
Yeah, sure. So clearly, cancer immunotherapy is an incredibly exciting area. Hot research being done. We are curing patients that were previously thought to be incurable using CAR T-cell therapy, using blockade of immune checkpoints that were developed by and put into clinic by people in this room. So very exciting. But we also heard from several of the panelists that not everyone benefits, and particularly some cancer types don't benefit from these therapies. And so we've learned a lot about both the successes and the failures of these therapies. And so it's already been alluded to several times that myeloid cells, other immune cells that may be in the tumor besides T cells, may play a role in preventing either T-cell recruitment or T-cell activation. And in fact, Dr. Sharma talked about all these other types of immune cells in your body. The tumor itself can have dozens of other types of cells, immune cells, support cells, muscle cells, blood vessels. All of those cells may play an important role in regulating T-cell activation or recruitment. In addition, we also heard that that CAR T-cells may have a hard time getting into solid tumors. CAR T-cells have worked tremendously in blood cancers, but there's a lot we need to still understand about their role in solid tumors. So Dr. Anderson is going to talk about new strategies there. When thinking about the myeloid cells or these macrophages and their complexities, Dr. Sharma alluded to the multiple types of macrophages. That's true. I'm a myeloid biologist myself, and so we know there's 10, 12 different types of macrophages, and we don't understand them. We don't know what they do in these solid tumors. We know they're suppressive, but we don't know enough about them to be able to target them yet. And so as a myeloid biologist, I'm always jealous of T-cell biologists because we know so much about T-cells, and so our lab is taking a step back, and many of the people working on myeloid biology, Dr. Mantovani and Dr. Marron, we're taking a step back to better understand the biology of these cells, and I think we're probably 10 years behind in terms of understanding myeloid biology compared to how much we know about T-cell biology. So those are some of the new things we're trying to do to utilize and target other immune cells within the tumor to be able to then activate and recruit T-cells, and then finding novel strategies to get T-cells into solid cancers.
Can you tell me a little bit about your work with breast cancer?
Yeah, so breast cancer is one of those cancer types that has some response to immune checkpoint blockade, but not tremendous successes yet. And so we do know, so breast cancer, pancreatic cancer, brain cancers, these are cancers that don't respond tremendously well to T-cell therapies such as the checkpoint blockade and CAR T-cells. But they are full of myeloid cells, these macrophages. And so we're focusing on trying to understand the biology and the complexity of these myeloid macrophage cells so that we can target the right ones for anti-cancer therapy. Because while macrophages can play this role where they can help the tumor grow and survive and inhibit T cells, macrophages can also, their normal function in your body is to defend yourself against bacteria and pathogen. They also can identify malignant cells and kill them. So we want to keep the good macrophages, but get rid of the bad macrophages. And we think if we can do that, we can better enable T-cell infiltration into breast tumors, as well as enhance their activation.
Macrophages don't attack tumors in the same way as T-cells
So it's a very different mechanism. T-cells attack based on a very specific signal that they receive from the target cell. Macrophages do need certain signals, but they can phagocytose, they eat things, bacteria, pathogens, based on sort of more common signals that they can then use to destroy foreign objects.
And can you talk to me a little bit about the surprises or the things that you found in your research that have potential?
Yeah, well, so first of all, that there's 14 types of macrophages in breast cancer, and that they're different between triple-negative breast cancer and hormone receptor-positive breast cancer. There's different types of macrophages. That was actually very surprising to us. We are finding novel strategies to target the most suppressive macrophages, and we think we can do that using new technologies. So we talked a little bit about CAR T cells, but we can also use CAR macrophages. So we can take the macrophages, or the precursors, the monocytes, out of the patient, the person, engineer them with these CARs, these constructs, and then use them to target cancer cells, to target macrophages themselves or other components of the tumor microenvironment. And so why we think this strategy might be efficacious is monocytes are in your periphery. These are the cells that they're in your body, and they get into tumors very well. I didn't mention this, but up to 50% of solid tumors can be macrophages. So clearly these cells can get into your solid tumors. And so we think that if we can design macrophages with these CAR constructs, we know they can get into the tumor, they can survive the harsh conditions, and we hope that they could be useful to target cancer cells for anti-cancer therapy.
Very cool.
One thing that is really important to know is that, Jen studies breast cancer, but her findings are actually applicable to all tumor types, because 50% of all tumors, or most tumors, are actually macrophages. And we're learning more and more that there's shared ways in which tumors really hijack your immune system between, the really hot tumors like melanoma and lung cancer, and then the tumors that don't respond to immune therapy like pancreatic cancer. And really by studying, her studies in breast cancer are not just specific to breast cancer, and this approach with CAR macrophages, it's not just specific to breast cancer. Also, we can really, study in depth what is happening in a patient who has brain cancer or pancreatic cancer or prostate cancer, and those findings we really can apply to the entire field of cancer immunology, because the more that we study this complexity of macrophages, we really see that these programs are really shared across all tumor types, and hopefully then we can develop therapies that we'll be able to use, across all cancers, the same way that, Jim's therapy was initially developed for melanoma, but now I'm using it in my lung cancer patients, weekly. So I think that, each of these steps, it's extremely important to develop therapies for breast cancer patients, because there are lots of patients that don't do well, but those, discoveries are really going to help cancer across the spectrum.
Can you talk to me about how you design your clinical trials and what you decide to focus on? Because you do a lot of work in early clinical trials. Tell me a little bit about how you pick a direction to chart your ship towards.
It's very tough. So we start typically by looking at all tumor types. So in a phase 1 program, a lot of the times we will be doing clinical trials in what we call all solid tumors. So basically anyone who has a cancer. Blood cancers are sort of a different beast, and typically different doctors are taking care of those. We just heard a lot about the leukemia and lymphoma programs. But we typically are trying to look across tumors because, again, if we have a good idea and something's working in mice, we don't know, A, is it going to work in humans, and B, what type of tumors is it actually going to work in? And so we want to enroll patients who have brain tumors and pancreatic cancer the same way we want to, look at lung cancer. But it's tough because, unfortunately, the reality is...
So you would take on all those different types of cancer patients for phase 1?
So yeah, so
Wow.
I'm a lung cancer doctor, so there's only normal patients I see outside of trials, but typically, over the course of the week, I'll see patients with all sorts of tumors. Typically in phase 1 trials, these are patients that have exhausted all of their standard therapies. So their breast cancer specialist will call me up and say, do you have a trial for this patient that has these characteristics because we've run out of good therapies? So
Right.
most of the time in those early phase trials, it's really, this is a last resort that many of our patients are pursuing, even though, most chemotherapy at that point also is not super efficacious. But we try it in everything, and then oftentimes we'll do a trial and we'll enroll patients with ovarian cancer and lung cancer and pancreatic cancer, and then all of a sudden I'll start seeing patients with pancreatic cancer respond, and we'll quickly jump on that and say, all right, let's try this in 40 patients with pancreatic cancer, and let's see if this is a real signal.
Because I was wondering, because presumably the number of people you have in a trial is limited. Then if they all have all different types of cancer, then figuring out how to compare those might be tricky. But if you see like pancreatic cancer or a different type of cancer responding well, that's when you start refining that.
Yeah, it's true. Like when we're designing these trials, before we even go into humans, we oftentimes will ahead of time say, all right, let's try this therapy in all cancer types. But based on the mechanism, we think that this will work best in pancreatic cancer and mesothelioma. But what I realize more and more when we're making these projections is we really don't know what we're talking about. And so we still have to learn a lot from these. And this really goes back to...
You presumably don't tell that to the trial patients.
Yeah, exactly. We try to limit that. No, we have to be very honest because, we're making decisions based on what we're seeing in mice and monkeys. We're not a bunch of inbred rodents. Yeah. And we're closer to monkeys, but most of our studies, we're not giving monkeys cancer the way we do with mice. But I think it really goes to the importance of immunotherapy programs like Pam and Jim have developed, and we have a similar one in New York, where we're really focused on giving these new therapies, but getting lots of blood samples and biopsies so that we can learn what's happening in these patients. I always, teach my fellows that, the greatest disservice you can do is do a clinical trial where you don't learn anything. And a negative clinical trial where at the end we just shrug our shoulders is just that. But if we do a trial, even if it's negative or even if most patients don't respond, if we can learn something from those samples,
right.
It's constantly pushing the bar forward.
And those clinical trials, you say therapies, are you talking about like combinations typically?
Yeah.
And so what sort of combinations do you look at?
So as somebody mentioned earlier, we have about 5,000 trials going on in the immunotherapy space. So there's lots of different combinations. Usually it's combinations with either PD-1 blockade or CTLA-4 blockade, the two FDA-approved drugs, plus something else. Sometimes we're developing brand new drugs that are targeting brand new molecules that are being discovered through the science that we're talking about. A lot of the times, one of the things I'm most interested in are learning ways to repurpose drugs. So taking drugs that we already have that are FDA approved and we know a lot about their toxicity and saying, hey, can we use drugs, sometimes even drugs like the obesity drug, the GLP-1 agonists that are actually showing very interesting signals in mice that they might actually modulate the immune system and help immunotherapy work better. So we have these drugs that we're already giving to millions of people or that we've given to millions of people in the past. Maybe we can incorporate those and use them in an intelligent way to help a subset of patients.
Right. Just to switch focus a bit, Kristin, tell me a little bit about your work because you are engineering these T cells. What are you doing? How do you do it? Tell me about your work.
Yeah, so I did my training in Seattle with Phil Greenberg, and he's one of the preeminent experts in engineering T cells to have a new T cell receptor. And so I went to his lab to learn how to do that, essentially. And when I was there, I was given the opportunity to work on ovarian cancer and try to get engineered T cell therapy to work for ovarian cancer patients. And we were able to, in mice, show that you can actually get T cells to recognize and kill ovarian cancer in that mouse model, but that there were a lot of hurdles, as Dr. Fox alluded to when he was up here too, lots of hurdles that T cells need to overcome in solid tumors in order to remain effective and keep fighting. And so what we do is we take concepts like what Dr. Allison and Dr. Sharma have done with checkpoints, and we look at the cell, we call it cell intrinsic changes you can make. So checkpoints, or molecules that send a negative signal to the T cell, we actually try to modulate that signal within the T cell itself, so that instead of having blockade of all the signals on all the cells, we target it specifically to the tumor-specific T cell that we've engineered. So, for example, we were talking about trying to get T cells into solid tumors more effectively. So when I was with Phil, one of the things that we worked on was making these synthetic molecules that essentially had the receptor that recognizes a negative signal or a death signal, but instead of sending a death signal to the T cell, which is one reason why a lot of T cells don't live in solid tumors, we made it so that that molecule would send a survival signal just to our engineered tumor-specific T cell. And so in our mouse models, that actually improved survival of mice, even if you didn't start treatment until they were at a really advanced stage of disease. And so that data is now going into building those tools for patients. So that's the kind of thing we do. We try to identify an off signal and rationally engineer our way around it so the tumor can't escape.
Right, and how translatable is that to other cancers?
Completely. So the molecules that we make, we can engineer them into a vector with a T cell receptor or a chimeric antigen receptor. You could put them into, for example, tumor-infiltrating lymphocytes. Maybe a patient had T cells in their tumor already that recognized a lot of different tumor molecules. You could put these synthetic agents into all those T cells and make all of those survive better. So it's a very versatile strategy.
You do a lot of work with patient advocacy. Can you talk to me a little bit about the importance of the patient in advancing this sort of immunotherapy?
Yeah, thank you for asking. I think this is one of the things I'm most passionate about when I wear my cancer researcher hat. I try to really think about, well, when I was a cancer patient, what was this like? And one of the memories I have that was most vivid was when I was diagnosed, I was a graduate student. So I was in my very early phases of learning how to be a scientist. But because I had been through my cancer journey, I was invited to be what they call a consumer reviewer. This is essentially a survivor or a patient advocate who gets to help review scientific grants. So I didn't have enough training to really review the science, but I had enough experience from my own personal journey to be able to ask questions about what impact would this have for patients. And I remember sitting on that grant panel, and all the scientists had already spoken about the grant, and it was my turn to talk. And I said, you know, this sounds like a really cool tool, but, you know, from my non-scientific understanding, won't this impact all cells in the body, not just tumor cells? And did the person write the grant in a way that they addressed that? And there was silence in the room. And then the scientists voted, and they all changed their scores, and that grant, I do not think, got funded. And so that's my memory of how a patient voice can actually change the conversation in the room, because patients bring a totally different perspective sometimes to thinking about science. And I assume everyone up here and everyone who's been on all of the panels so far has found team science and multiple brains working on a problem to be more effective than one person trying alone. And I think when you bring very outside the box thinking and different perspectives to a problem, you're going to come to a solution much faster.
We have a surprise for you. The person who went for that funding is here with us in the room today.
Oops.
Alberto, let's talk about macrophages. What is a tumor-associated macrophage? I know kind of Jen kind of touched on that already, but tell me about your work and your area of research, please.
Yeah. Ages, well, the observation that was mentioned, we have known for ages that there are plenty of macrophages, host cells within tumors. So the question decades ago was, what are they doing there? And we had hints that more macrophages there would mean worse prognosis, which was intriguing. The immune system is very complicated. It's as complicated as the central nervous system. the central nervous system, the brain, and the immune system are the two most complicated and the two heaviest systems in our body. And macrophages are part of the first line of resistance. We believe that the first line of resistance, which we call innate immunity, takes care of 90% of the encounters with bad guys that we have each and every day. So what are those cells? The question at the time was, what were those cells doing in tumors? And in general, they were believed to exert anti-tumor activity. And at the time, I found that they were actually promoting tumor growth. One of the tumors that I studied was actually human ovarian cancer because it was easy to get. And that's the emphasis, which human cancer, we need to study human cancer. And of course, we had it wrong. We got it wrong because we were talking of macrophages, period. And Jen said, well, there are plenty of variation on the theme. And then we are discovering that they have checkpoints. For instance, why macrophages, why they don't eat red blood cells, erythrocytes? Well, because there is a signal on red blood cells. They say, a don't eat me signal. And some tumors have upregulation, some of the lymphomas, the non-Hodgkin lymphomas that was mentioned, they have upregulation of the genes that encode and sends a message, don't eat me. My count was that there are 13 checkpoints in macrophages. My count today is that there are 14 because we have another one coming up. The hope and expectation is what was said originally, was said by Padmanee, that we would combine myeloid targeting therapies together with T-cell targeting therapies. And the perspective, I mean, I'm an oncologist by training, so I've seen combination therapy grow and save and save patients. So I think that we are in that situation, combination therapy.
So, and this is maybe a really stupid question, but are you talking about checkpoint inhibitors for macrophages as well as other cells?
Yeah, well, there are trials ongoing with some hope for that they may increase, and there will be more coming, and we respect data and we wait for the data.
Okay.
And then there is, and Jen mentioned that the hope of doing, these cells are attracted by tumor. This actually was one of the intriguing and unexpected discoveries in my early life. Why should tumor cells attract a macrophage? And
do you think
that's what Jen was mentioning, saying that these cells are professionally entering the tumors, and perhaps we can take advantage of.
Do you think that's a very promising avenue for research? This is question of why we're finding or how macrophages get into solid tumors? And
well, I think, of course, I mean, I've been in love with macrophages all the time. So the answer is obvious.
Yeah.
If I may. I mean, these are corrupted policemen, but I think we can re-educate.
I think the point that the majority of the immune cells in the tumor are macrophages really underscores the fact that they must be doing something there. And I always when I'm talking to patients, I always sort of emphasize the complexity is we have all these different immune cells, and there's always the good guys and the bad guys. So we want to find therapies that either get rid of the bad guys or turn the bad guys into good guys. And that's exactly what checkpoint inhibitors are able to do. Checkpoints are really just stop signs that we're trying to tear down so that your immune system does its job. So if we can figure out a way, I mean, it's amazing that we have these therapies that harness 10% of the immune cells, which are the T cells in the tumor. If we can harness 50% of them, which are macrophages, and turn them all into cancer killers, we could cure everyone's cancer.
Very good.
I drank the Kool-Aid, so.
I do want to have time for questions. Maybe will we open for questions? Is that okay? Does anyone have something they really want this audience to hear? Because I do have more questions, but I feel like in terms of time, I would like them to do that. Is that okay?
Yeah, that's fine.
Okay.
Can I mention one?
Yes.
I think we have a gender issue. So we know that
yes, I wanted to ask you
immunotherapy, there are differences between females and males. For instance, non-small lung cancer is one example. Checkpoint blockade immunotherapy works less in females. We have less female patients entering trials. The immune system of females is different in many respects from the immune system of males. We have looked at the immune system in 2,500 patients in silico, non-small cell lung cancer, males versus female, profound differences. So I think that, and this is why, I mean, I'm supported by a charity in Italy. I've been supported for 50 years by a charity in Italy. So I think we have to address and to have a gender perspective also for non-gender related tumors. And perhaps charities could help.
Yeah, that's actually a really good point. Yeah, and thank you for reminding me of that. We have, where is Aidan? Aidan, if you could come up and help us out. So if you have a question for our panelists, including all of our speakers, Jim and Pam and everybody, if you have a question for them, you just raise your hand and we will get a microphone. If you can wait for the microphone to come to you before you ask your question, that would be great. And then Aidan will help me. You just say, you point at the person here. Well, you guys can stay on stage actually if you don't mind, because there might be questions for you that we hit you. But so if you have a question, raise your hands. The time is now. Yes. No, it's okay. You're fine.
Hello, everybody. Just want to thank you for putting on such a great event, Breakthrough Cancer Research. My name is Michael Rinn from CLL Ireland. My wife is a CLL patient since 2011. She was diagnosed when she was 39. And just to echo what Owen and Larry Bacon had said, like it's such a struggle in Ireland. Less than 2% of cancer patients can access clinical trials. I don't really want to go on a long speech, and you don't want that. I don't really have a question. It was more a comment just to thank you for putting this on.
Yeah.
Also, tomorrow, September the 1st, kicks off World Blood Cancer Awareness Month, and it's also World CLL Day. And just to let people know that the first patients in the world to get CAR-T were actually 3 CLL patients back in 2010. And 2 of them patients went on beyond 10 years, which they reached that cure. So that's, we kind of have to kind of honour the patients.
Absolutely, absolutely. Just on that, because we'll get to this lady here, then if we get a microphone to her. Just very quickly, Owen, you know, the talk is, you know, we've got to speak to our politicians to get this moving along. Aren't the politicians already on board? Like, and why is that? Because it's very difficult. But like, what do you mean no? Like, why would a politician not be on board just for money? That can't, like, it's very difficult to answer. If I was a politician up here and he said, well, look, we haven't got the money, is that actually the response? Or do they not understand the opportunity, or do they feel like they're too far spread?
It's a lack of awareness. I remember CAR-T actually when it first started rolling out around Europe. All of the countries in Europe had CAR-T, and Ireland didn't have CAR-T. So it's a real credit to, it's a real credit to the clinicians here, and to hear the fight that they had to put up to get CAR-T into Ireland. I do remember when the CAR-T centre had opened down in James's Hospital, and I have a friend advocate over in Spain, and he didn't know about that CAR-T centre opening, but she had actually tweeted that great news from Spain. We've opened 18 CAR-T centres on top of the 14 we have already.
Yeah.
So that's...
Which says a lot, right? Owen?
Yeah, I totally agree. So at that stage, Larry and ourselves and the Children's Hospital were sending some of our patients into the UK for CAR-T. And this was through a time when we had COVID, pan-global COVID. It was horrendous. So the issue is, the nub of the issue is that there's a failure to fully understand what we're doing, but also it's a funding mechanism as well. And that's why I was saying with the new program, Impact, it'll bring partnership across industry, academia, hospitals, and it'll lower the amount to treat these patients in the long run. We just need to engage them even more.
Okay, great. There's a lady there at the back. Oh, have you got a microphone? Oh, no, I thought you had one.
Go ahead.
No, please go ahead.
Okay. Hi, everyone. My name is Angela Steen. I was diagnosed with melanoma in 2018. And in late 2018, the surgeon who operated on me believed I had about 6 months. I was then treated with immunotherapy, with much thanks to Professor Allison here. And I was 35 in 2018. I just turned 42. So you're my hero. And yeah, thank you so much. I do have a question, though, and not specifically addressed to one of you, but if anyone is interested in responding, it's a little bit of a cliche. But what is one thing, if you woke up tomorrow morning and it was resolved, would make your work and your agendas much more effective?
Well, let's start off with Jim, perhaps, for that one, if that's all right, Aideen. It seems he's travelled all this way, our Nobel laureate. Is that all right? So, Jim, what one thing, if you had resolved to tomorrow, would it change the landscape of your work?
It was already mentioned the way of making the myeloid cells help the T cells cure all cancers.
Pam, do you have a different answer to that?
Yeah, I think it would be very important for the clinical trial design. How do we start looking at clinical trials more for immunotherapy settings, and how do we bring that into earlier stage disease for more of the neoadjuvant trials?
Great. Larry, maybe if we have another question, we might go through, because it's a good question. Thank you very much. If you have another question, we'll go to you. But maybe, Larry, what's your response from an Irish perspective?
I guess from an Irish perspective, it is that we get that we have access to an improved access to clinical trials.
Yeah.
We have access, for our patients, we've access to more beds, access to more funding to open 7-day wards, to make all of our lives a lot easier and more importantly, the patients' lives.
Okay.
Yeah, on a similar vein, I would say for hospitals to understand the importance of research.
Yeah.
I think research very much in an everyday clinical context sounds like something that's aspirational. It sounds like an extra. Oh, you'll do the day-to-day things, and if there's extra, you'll do research. I think there's a fundamental lack of understanding at a government level that research is care, that research is treatment, that research is actually hope. They think it's an extra. And I do think that the patient voice is far more powerful than any of our voices to explain to the powers that be that it was research that allowed them to live.
Very, very well put. We'll go to this lady here, unless you guys, yeah, we have another question here. This lady, yeah.
Yeah, hi, Miriam Staunton, also similar to Ange, was diagnosed with melanoma in 2018, the lucky recipient of immune checkpoint inhibitors, and they worked with significant toxicity, lifelong impacts as well. But I guess for me, two things, apart from reiterating Michael's point of thank you so much, it's a brilliant lineup and it's unbelievably interesting. Clinical trials, we are poor at getting them to this country, and it is, one of the few routes we have to accessing drugs early. So I really find it difficult when people mention that, getting people to participate. Getting people to participate is not the issue. It's having access to the trials is the number one issue, and a well-designed trial will be oversubscribed because patients will want to get on it. In terms of accessing drugs and approaching the politicians, I'm all ears as to what we can do better. I had to wait to progress to stage 4 before I could access treatment because it wasn't funded at stage 3. We're hearing the same story around, not for this cancer, we have a CAR T for this cancer, but not that cancer, even though we know it works. So research is important, but like this is invented and we can't access it.
Yeah.
Because why? Because we're dragging our heels getting stuff approved or EMA approved. Why have we got such a long process and this pretense of getting patient, we spend months putting together patient viewpoints on this, and then you're told, oh, well, it doesn't compare well with the standard of care. Yeah, well, it's not going to. From a cost perspective, standard of care is no treatment. I mean, so what can we do together, as clinicians, as patient advocacy groups, to actually, bring this to a head and say, because we are a rich country, we are spending a lot of money on healthcare, so why can we not access it? And we're now seeing this wedge happening where treatments are now more and more of these new treatments that are available to private patients and not available in the public system. And we have, that's just so inequitable that we'll have patients in the country that can't access and patients that can.
Yeah, okay. There's a gentleman at the back.
Hi, my name is James Hastings. I'm a cholangiocarcinoma stage 4 patient. Luckily for me, well, it was lucky, I'm MSI high, DMMR, MTB 17 mutations. Very thankful to Dr. Allison. I went on the CTLA-4 PD-1 Ipi-Nivo. It worked incredibly well. Now, having said that, I had to go overseas to get that treatment because it's not accessible. MSI high patients are not available to access these drugs here, which is absolutely criminal. But unfortunately, after 9 months of maintenance PD-1, maintenance Nivo, I've now got recurrence, progression of my cancer again. So I've kind of got a question. Would you suggest I re-challenge it with a CTLA-4 PD-1 or a CTLA PD-L1 as a very good chance of success? Now, it's a cholangiocarcinoma, so there is no documentation in the world I have found of anybody re-challenging MSI high with a CTLA PD-L1. It'd be great if anybody could give me any insight onto it.
Yeah, I think Pam is going to give you some. We won't go too much, if you don't mind, not too much into individual, but like we will definitely have someone to chat to you about it. But Pam, do you want to just comment?
Yeah,
on
I
that?
think if it's still MSI high disease, absolutely that immunotherapy and Ipi-Nivo again can be tried. I would not take that off the table. I'm so sorry to hear that MSI high is not approved in this country to receive immune checkpoint therapy. It's one of the few settings where we know patients do very, very well is the MSI high status.
Yeah, I can see Jim and Pam, when you were talking about, they were just kind of shaking their head in disbelief. Liam?
Yeah, I just want to again echo what Michael and others said. It's a fantastic event, and it's great to see and hear from all the different speakers. I'm just wondering, in terms of, like, a lot of discussion is around cost, but also this issue of efficacy and the modest percentage of patients which respond. Again, one way around that is better selection of patients in terms of upfront, and there was initial interest, I think, in terms of trying to find biomarkers, predictive biomarkers in the immunotherapy space, but it's almost like it hasn't really fully penetrated properly into that whole field in comparison to more targeted therapies. So I was just wondering, what do people more generally feel about the developments in that space? Because the classic ones are pretty poor, and there's lots of emerging ones, but it doesn't seem to have penetrated into the clinic. And I think that's kind of frustrating, because it sounds like, okay, it kind of might work for you, so therefore you give it to everybody, and that leads to this significant cost burden.
Yeah, Pam?
It's an important point, and that's why I was just saying MSI high is actually one of the best biomarkers for selecting patients. So if you have an MSI high status, you should be getting immune checkpoint therapy. But you're absolutely correct. We don't have great biomarkers, and that's because your immune system, as I mentioned, is individualized for you. And so we have to start to understand what we want to look at in these individual settings, because in lung cancer setting, it might be different biomarkers than what we look at in bladder cancer settings. So I think these are things that we're working on now, but I don't think it's, from an ethical standpoint, I would have a very difficult time telling a patient with MSI high that they would not get immune checkpoint therapy.
Yeah. Unfortunately, MSI is a place where we do see amazing responses, but then unfortunately there are certain patients where it stops working or where there's relapse. In most of our patients, they don't benefit from immunotherapy. So to your point, most of my lung cancer patients will die of their disease. And so it really underscores the importance of investing in those biomarker studies, which are the translational studies that Dr. Sharma had mentioned. And it's why we really should encourage patients and hospitals to invest in these sorts of resources where we're collecting tissue and blood blood, at baseline and while patients are on treatment, so we can identify those biomarkers. When it comes down to it, for-profit companies are not going to invest in identifying a smaller population to give their drug to. They want to give their drug to as many people as possible. So it's really reliant on the patient advocacy groups and the scientific community to identify those biomarkers to really refine the treatment algorithm. So we give these therapies to the people where they will work, and the people that it's not going to work, let's not waste their time and energy and potential toxicity with a therapy that's not going to be effective. So,
Jen, you were nodding furiously there. Do you want to add to that? And then Kristin.
Well, certainly on treatment.
One second.
Baseline.
No, go back to
Baseline and on-treatment samples analysis are incredibly important. I wanted to shout out to the foundations such as Breakthrough for Cancer Research. These are the foundations that are funding this type of work. We cannot get this funded by the federal government. These are exploratory studies. They're sometimes high-risk studies that cannot be funded in other ways. So we need funding agencies to help us investigate and do these deep dives into tissues to correlate what might be in these baseline, these pre-treatment biopsies that could predict whether a patient's responding or if they're not responding or if they have toxicities too.
Right. Kristin?
That this is actually an ongoing discussion in the Society for Immunotherapy of Cancer, and we're actually at the moment working on a white paper or a recommendation article that lists biomarkers for immune response, non-response in tumors. That way we have much more comprehensive suggestions for the whole field in terms of what should we be looking at all stages, and what should we try to make mainstream in terms of the biomarkers that we're regularly looking at for this exact reason.
Byrne, did you want to add to that?
I think that covered it.
Okay, great. Do we have another question? Yes, in the middle there.
How you doing? I'm Eileen. Thank you to all the speakers, and it's an honour to be in the same room as a Nobel Prize winner, so well done, Jim. Sorry, because I came up with the four speakers about it's ideal for early intervention with the immunotherapy, but I was just wondering, does any speakers see a future for metastatic cancers? And I know because lung cancer came up a lot earlier on, I'm just saying specifically cancer that has metastasized to the lungs, or is that a no-no at this stage? Am I too premature with that
Alberto?
question?
No, I want to repeat something that Pam said, namely that there are sites of metastasis which pose specific challenges. The liver is one. The liver, in a way, is born as a what we say tolerogenic site, and of course, as clinicians, we are challenged with specific. We have specific challenges with specific sites. We are trying to understand the diversity of the immune system in different organs. It's born different. The liver has specificities in terms of the immune cells that inhabit the liver, the bone, which of course, and I think that this is one of the challenges that we have, namely to understand the complexity of the immune system, or metastasis at different anatomic sites. And I want to add one more challenge that we have, for which we need funding of high-risk research and charitable and philanthropy, at least in my case. And that is, if you take, our genome encodes for words, which we call proteins. And there are about 15% of the words encoded by the genome. We don't know the meaning of the words. And some of them are there in our immune cells. And so we need to understand the meaning of the words and the meaning of the words in the different organs and of the cells. And this is one, I think, is one of the challenges that we have when we are confronted with metastatic tumors.
But
Pam, did you want? Oh, yeah, Pam. Go ahead.
I want to say, but the most of the therapies are approved for the metastatic disease setting. So the immune checkpoint therapies are approved in stage 4 metastatic disease settings. What we're trying to do is get them approved in earlier stage disease settings, because we think if we give the therapies earlier, before the patient has chemotherapy, before the patient has disease that's spread elsewhere, then we'll get better responses. But if you have metastatic disease that went to the lungs, for example, if you have bladder cancer that metastasized to the lungs, you should be able to get immune checkpoint therapy because it's approved in that setting. If you have kidney cancer, renal cell carcinoma that's metastasized to the lungs, you should be able to get immune checkpoint therapy. It's approved in that setting. So I just want you to, to say that yes, it's approved for metastatic disease.
Because I was just wondering, like, if just say it was breast cancer that has metastasized, tended to that, but the cancer in the lung is new. It's just...
Yeah, so it's not called lung cancer then. It's just breast cancer that went to the lung. Yes, exactly.
Okay.
I would say that, as Dr. Sharma mentioned, most of our therapies are developed in the metastatic setting, and this field is moving so fast. So 2 weeks from now, we're all going to be in Barcelona for the European conference, and there's going to be transformative data that's presented there. There's novel immunotherapies that are getting approved every month, and so there are lots of therapies and lots of reasons to hope. Obviously, we need to be working to support these trials and the science, but this is a very exciting time for the field of immunotherapy, and I think we're going to see, dozens of therapies approved just next year.
Thanks, Tom. Yes, over here.
From Ireland, so obviously I'm a massive fan of immunotherapies. It's keeping a lot of people that I've grown very close to alive. I just like to draw on what Mariam said earlier. I think the most hated word I have at the moment is cost effectiveness. Because there is so much evidence for the absolutely phenomenal response in deficient mismatch repair cancers to immunotherapies, not only in the metastatic setting, but also in the neoadjuvant setting, where I think we're on the verge of a new era of medicine of organ preservation for this group. And currently, one of the big stars seems to be dostarlimab. And it is currently approved under the European Medicines Regulatory Authority for the treatment of advanced and metastatic mismatch repair endometrial cancers. It is currently up for review in Ireland after being previously told no in 2021 because it costs too much. I think we need to get rid of the idea of what cost effective is when you're talking about people's lives. When something is approved already and shown to be effective, how can the most wealthy country, as we call it, in Europe say, sorry, no, you can't have that. Your friends in France can have it, your friends in the UK can have it, but you can't have it here. And I think that really hits home a lot in the comments that we, the patients, we have a responsibility to step up and fight that fight. I think one of the best bits of advice I ever got starting off as an advocate was when I asked a surgeon if he would, this was like 7 years ago, I'd first heard of immunotherapy following Tim Ellison's work actually. I knew then that this was going to be game-changing. And I asked the surgeon, can you try and, get this immunotherapy in here for colorectal patients with Lynch syndrome? And he said, well, to be honest, you have more power than I do. And in that moment, I thought he was fobbing me off. And I realised over the last years, all these scientists, doctors, all the wonderful professions, you know what works. You're talking to people that are outside of that spectrum, government officials, people with the purse strings, and they don't really care. All they care about is the cost effectiveness, and you're just another doctor or another professional waving a flag about something that they can happily ignore. But they will listen to the patients, not because it's the right thing to do, it's because public perception is everything.
Yeah.
And right now, like I said, dostarlimab is up for review again. For advanced endometrial cancer here in Ireland for people with mismatch repair deficiency that have exhausted all other options. So cost effective is a word that shouldn't exist. So if everyone sees it online, can back us. And to be honest, the only reason we know about it as an organisation is a wonderful oncologist in Galway, Michael McCartney, who tweeted about it. Because I looked at the last review for that drug, for this indication, and it said no patient submission was put forward. We never knew. We never got the opportunity, and it shouldn't come down to chance. So I agree with what everyone is saying here. There's so much to be done. We need so much more stage 1 trials, like the vaccines for Lynch syndrome that are happening over in the States. I thought were a fairy tale 7 years ago when the first, almost like whisper urban legend came about. I thought maybe it's something I might see in my lifetime. It's in 4 sites across the States. Why don't we have it here in Ireland? Because I have 300 Lynch syndrome patients in a group right now
that are not like build it, we will come. They're there. They're at ground zero. They just need somebody to open the door. And they're so motivated because they're not just thinking of themselves, the generational effect of the gene on their family. They're thinking about who's coming after us. And I think everybody in this room, no matter what cancer you're advocating for or what treatment, everything we have is off the backs of everyone before us. So we have to play it forward. And I think whether it's going to be for lymphoma or whether it's going to be for a breast cancer, does it really matter at the end of the day when it's people's lives? I think we all need to kind of bolster it together. It doesn't, I'll help you fight for, for CLL. You help me fight for Lynch syndrome. Yeah. And I think, us patients, we can always have this thing like, oh God, the professionals don't talk to each other. Like, how come they're not listening to what they're doing in that country? Everybody is working in silos. Us patients, we're just as bad. And I'm realising that. And I think we all need to kind of look at the example that you're doing, what you're doing here today, coming from all over the world to try and get this message out there, to raise awareness, to support each other and support the patients. I think us patients and organisations need to back all of you to the same extent.
Yeah, and we've seen the impact of high-profile patients, how they become front-page headlines. They're on the Late Late, they really advance, and they embarrass the politicians a lot of the time. Yeah, it comes down to literally just, although it is often, a burden on the patient as well when they're already going through a lot, but we can see the power of it. And I think, a really interesting way to think about it, that the patient has more power sometimes than the clinician. Great to hear from patients, and I think maybe that's probably the second last question, because this lady has been waiting quite some time. And then we're going to hear from Joanne. So you have the last question, madam. Would you go ahead?
Sorry, I just wanted to ask, just in regards pancreatic cancer. I know it is one of the harder ones to treat with immunotherapy, but my mom has pancreatic, and we're going back and forward to the oncologist, and there's no mention of clinical trials, genetics, biomarkers, nothing being mentioned. It's just every time I ask something, it's no, there's no real why. And I'm not giving out. I know it's really difficult cancer to treat. I understand that. But I'm just wondering, where do I go now? Like, do I go clinical trial? How do I get onto that? Like, can I go? Is it better for me to go abroad now? Like, and where do I even start with that? It just seems to be a bit of a minefield of information online. And yeah, I guess I just...
Look, I mean, I can totally understand why you'd be here and ask that question. Is there someone on the panel who can help? I mean, I know we don't talk too much about individual patients, but...
So I'm sorry to hear that they're not doing any sequencing. One of the things as an oncologist, is treating patients. The thing in this day and age, every patient should have their tumor sequenced. Every single patient should have their tumor sequenced, because we have so many therapies out there if you find specific targets that are approved. For example, if your pancreatic cancer is MSI high, they should have immune checkpoint therapy. But you're not going to know that unless you do the testing. There are all these other targeted therapies that have now been approved based on specific mutations. And it doesn't matter if the mutation is in a bladder cancer, a kidney cancer, or a pancreatic cancer. If they find that mutation, the drugs are approved. And maybe not here, because I don't know enough about what's approved in Ireland, so I apologize for that. But absolutely, if you can, and if it's not here, if it was me, I would go somewhere else than for my loved one.
Is it somewhere from an Irish context you can say anything on this? Thanks, Jarushka.
I think it probably, for confidentiality reasons, maybe you and I should have a chat afterwards, and we'll take it from
that would be great. Thank you very much, Jarushka. I was hoping someone would say something. Okay,
can I give one resource?
Yes, Kristin.
So again, I don't know if this one is useful for those who are in Ireland specifically, but the NCI in the United States has a hotline that you can call, and it's like 1-800-4-CANCER. And they're staffed with people who will do the clinical trial searches for you. So you can be a patient, you can be a provider, and you can call with information and say, here is what I know about the tumor. What clinical trials would this person qualify for? And then they will list every trial in the United States that's open and say, these are all the ones, and here's who you should contact to figure out if this particular patient you're calling for is eligible. So if you are open to going abroad, that's a resource you can use to try to potentially find trials that might be relevant.
Thank you so much for that, and I'm sorry to hear your news. I hope you do find some help, and thank you, Jarushka, so much for volunteering to chat to you later. Look, it's been really fantastic to get people together, to hear patient stories, to hear about Hope on the Horizon. It's been really wonderful having you all here. So thank you so much to our panel. We're going to hear now from Professor Joanne Lysaght, who's going to finish off the evening. But just before she comes up, thank you so much to the hotel, McCrea and the Vid Academy for their work here. Joanne, thank you.
Thanks, everyone. My job, I was told, was just to say a load of thank yous, so that's what I'm going to do. But really, to, I suppose, explain where this meeting came about, we have to go back to the Irish Society of Immunology, which I'm the current president for. We, in our infinite wisdom, bid a few years ago to host a European Congress of Immunology, which is the largest immunology conference in Europe. And we have the Congress president with us here, Professor Ed Lavelle. So that was a week-long meeting that's going to happen next week. And as president of the ISI and Aidan Ryan as secretary of the ISI, we're both cancer immunologists, so we had a very vested interest. So we decided to have a cancer immunology meeting that's going to take place tomorrow. And for that, we invited world leading experts, both national and international leading experts, to Dublin. And we were absolutely honoured that they all agreed to attend. And then we were approached through the infinite wisdom of breakthrough cancer research. And they said, having all these experts, on our doorstep, cancer patients in Ireland will benefit from that. And so we've worked with them. They've been an absolute pleasure to work with. And you can see the benefit of, I suppose, the network Breakthrough Cancer Research have and the expertise that they have in bringing these scientific and clinical experts in front of cancer patients. And really, it's such a joy for us. And as cancer immunologists, I've thoroughly enjoyed this evening. And I always learn loads, especially when there's patients in the room, because it is such a different perspective than what you'd have when you're just dealing with scientists or clinicians. So my list of thank yous. First of all, I'd like to thank Jonathan for emceeing the evening. Again, it can be a very emotive and complex area. And for someone, I think you did an absolutely amazing job. So thank you very much. So I'd like to thank all the team at Breakthrough, especially Orla and Linda, who've been working really hard. I think when an evening goes really well like this, it seems that there's not much to organising, but there's a lot of organising in the background. So to the whole Breakthrough team, thank you so much. Obviously the hotel here for hosting us, and then moving on to our absolutely stellar lineup of panellists. It's honestly such an honour to have you all here, and just thank you very much for giving up your time, and we hope that you've enjoyed it as well. And lastly, I'd like to thank you, the audience, for coming. As I said, without you, this is just us talking to ourselves, which is a scientific meeting, and no one really benefits. So I want to thank you as well for coming out and taking your time. We hope that you've benefited from this evening. So safe home. Thanks very much.